Yeah, this slide will give a comparative account of somatic cell gene therapy and germline gene therapy. Okay, in somatic cell gene therapy, therapeutic genes are transferred into somatic cells, whereas in germline gene therapy, the genes are transferred into germ cells. So in somatic cell gene therapy, genes are put into bone marrow cells, blood cells, skin cells, etc. Whereas in germline gene therapy, genes are introduced into eggs and sperms. Somatic cell gene therapy here, the characters will not be inherited to later generations, will not be passed on to the successive generations, whereas in germline gene therapy, it is heritable and passed on to later generations. At present, all researchers, prefer to correct genetic defects in somatic cells, and for safety, ethical and technical reasons, germline gene therapy is not practiced right now. Then, this slide again will just schematically tell you the differences between germline gene therapy and somatic gene therapy, wherein in somatic gene therapy, the somatic cells are targeted and not the germline cells, whereas in germline gene therapy, all cells are targeted including germline cells. Okay. Then there is one more approach called embryonic gene therapy. Here, the permanent transfer of a gene into the cells of an early embryo just after the sperm and egg unite, is done. So, the moment zygote is formed, okay, the bad genes are replaced by good genes. Embryonic gene therapy refers to the permanent transfer of a gene into the cells of an early embryo just after the sperm and the egg unite, soon after fertilization. Whereas germline gene therapy, as I have told you, refers to the permanent transfer of a gene into sperm or egg cells. Whereas in embryonic gene therapy, the gene is transferred to a zygote soon after the union of sperm and egg. Whatever the case may be, in both, the delivered gene would become a permanent part of cells in the resulting adult. Okay. Then, in embryon gene cell therapy, what is done? What they do is, here is a patient with the genetic defect and biopsy is done and the patient's cell sample is taken, and then at the same time you have to take an enucleated oocyte, that is oocyte without the nucleus, and then the nucleus of the patient cell is transferred to the enucleated oocyte, and later this oocyte is done in vitro culture. Okay, in vitro culture of this oocyte with patient's nucleus containing non-functional gene is put to in vitro culture, and you will get a blastocyst, early stages of embryo, blastocyst out of it. From the blastocyst, you can retrieve embryonic stem cells, which are totipotent, means they have potential to differentiate into any type of cells. And then you should have autologous human embryonic stem cells with genetic defect. You can do genome editing, that is homologous recombination or gene therapy, so that you will get human embryonic stem cells with corrected gene defect. And they should be subjected to in vitro differentiation. That means they are allowed to differentiate under laboratory conditions, and these cells are transplanted back into the patient with the genetic defect so that the patient becomes normal. The bad gene will be corrected. This method is known as embryonic gene therapy. Okay, then. So this is another slide which gives you the gist of embryonic stem cell mediated gene transfer. What is done? Isolation of totipotent stem cells from embryos or blastocysts, and into these cells you are going to insert the desired gene, and recombinant cells are incorporated into the host embryo, and that's how the defect is corrected. Okay. So these are three approaches: somatic cell gene therapy, germinal cell line gene therapy, and embryo cell gene therapy. Apart from that, you have types of gene therapy. There are two types. Okay. There are two types of gene therapy. One is ex vivo gene therapy, and the other one is in vivo gene therapy. Ex vivo gene therapy involves transfer of genes in cultured cells like bone marrow, and these bone marrow cells which are cultured are reintroduced back into the patient. So this happens outside the body of the patient. This can be applied only to selected tissues as like bone marrow cells, whose cells can be cultured in the laboratory. All cells, it's not possible to culture all cells in the laboratory. Conveniently, which cells if you can culture in the lab, so ex vivo gene therapy can be done only to such selected tissues, not to the other tissues. So what are the steps involved in this? So this is the schematic representation of ex vivo gene therapy. Now, you have to just isolate the cells with genetic defect from a patient. These are the cells which are isolated from the patient. Then you have to grow those cells in the culture. Then you have to introduce the therapeutic gene into these cells to correct the gene defect, and you have to select the genetically corrected cells which are called stable transformants, and you have to grow them. This is what is done in ex vivo gene therapy. Okay, here is the patient. Cells have been isolated with the defect. They are cultured, and you have to give them the therapeutic gene, okay, to correct the gene defect, and then those transformed cells are put back into the patient. So transplant the modified cells into the patient. The procedure basically involves the use of patient's own cells for the culture, genetic correction, and then return back to the patient. As a result, this is not associated with adverse immunological responses after transplanting the cells, because patient's own cells are taken. This therapy is efficient only if the therapeutic gene or the remedial gene is stably incorporated into the genome of the patient and it is continuously expressed to produce the functional proteins. This can be achieved by the use of good vectors. Okay, there are success stories for this. ADA, I was telling you, adenosine deaminase deficiency resulting in SCID, that is severe combined immunodeficiency syndrome. This has been corrected by ex vivo gene therapy. Then hypercholesterolemia, that has been corrected. Then Leigh-Nahan syndrome, hemophilia. These are all the genetic defects, diseases caused due to genetic defects, and they all have been corrected by ex vivo gene therapy. So these are the success stories. Then the another type is in vivo gene therapy. This involves directed delivery of the therapeutic gene into the target cells of a particular tissue of a patient. It is done inside the patient's body. Okay. Many tissues are potential candidates for this, like liver, muscle, skin, spleen, lung, brain, blood cells, etc. And here, gene delivery can be carried out either by viral method or by non-viral vector systems. Both the way you can do, though right now viral vector systems are used. And the success depends on efficiency of the uptake of remedial gene, then intracellular degradation of the gene and its uptake by the nucleus, expression capability of the gene, and many genetic defects like cystic fibrosis, hemophilia have been corrected by this method. So what exactly is done in vivo gene therapy? See, the copies of therapeutic gene, that means good genes, are inserted into either viral DNA or into liposomes or in the form of plasmid DNA, and later the genetically altered DNA is inserted directly into the patient's body by cell-specific direct tissue injection, and once inside the body, the inserted DNA is incorporated into the cells of that particular tissue, and these cells will now produce good functional proteins by the good genes, and therefore the patient becomes normal for that particular deficiency is concerned. Okay, this is what is done in vivo gene therapy. Right now, modified liposomes are not currently used. They are making use of viral systems only. This plate gives a comparative account of in vivo gene therapy with ex vivo gene therapy. So here is the patient. Therapeutic gene is directly introduced into the body of the patient. This is a less invasive procedure. Directly tackle the proliferative tumor cells to inhibit cell division. But the drawbacks are require the targeting of tumor cells, possible immune responses to gene vectors. Coming to the ex vivo gene therapy, where the cells are removed from the patient, they are cultured, and then into them therapeutic genes are put, that is, cells are infected with good genes or therapeutic genes, and these transformed cells, cells with therapeutic genes, are put back into the patient's body. Plus points are selection of target cells and immunocompatibility. But the negative points requires an invasive procedure for solid tumors, and non-proliferative altered tumor cells will not outgrow over highly proliferative tumor cells. So this is the difference between in vivo and ex vivo. It just briefs you the comparative account. My dear students, thank you for your patient listening. Any questions, do contact me. Till then, stay blessed. Thank you.